So in this video I'm going to talk about the chemistry Nobel Prize for protein structure prediction. Okay, which uses AI, and I actually think is being overhyped, and I'll explain that in the following video. So this article is published in Nature last December, and it's about the accuracy of structure prediction by AlphaFold 3, the third version of it. And there's a section in it on the model limitations. See, this is limitations of AlphaFold. So the three versions of it, AlphaFold 1 2018. So it's only been good for really best at predicting the structure of monomeric proteins. Where it's just basically single bits of, like, a hemoglobin is made of 4 parts. It's been good at predicting one of them. That's what that means, I think. Rather than the biologically relevant complexes, a hemoglobin exists as 4 parts, and you got actin, myosin, muscles exist sort of as basically two more parts, and you got a various other proteins in the body, which in plants and animals, which consists of multiple subunits. As I said, proteins with disordered regions are actually predicted with low confidence, so lower percentage success. So something might be only 30% accurate, or rather than 90% accurate. AlphaFold 2 has not been particularly great at predicting the effects of mutations in proteins. The model relies on evolutionary information. You might get 2 identical proteins from very different unrelated species, which have completely different ancestors. So that's an example of convergent evolution. Or you might get two species from exactly the same ancestor, but where the enzymes are completely different, say, or proteins are completely different. So it does require evolutionary information, and proteins which are more unusual or unique have low success. It's apparently not very good at producing multiple native conformations of proteins. What did I think this was the other day? I think this refers to probably enzymes, where you get a structure when you got substrate bound, a substrate with the product bound, and the third substrate when you get the substrate half bound. Or when you get hemoglobin, each molecule of hemoglobin requires 4 oxygen atoms, molecules, not atoms, molecules, O2. And so as each oxygen molecule binds in turn, so one oxygen molecule binds, that leads to a second one binding, which leads to a third one binding, which leads to a fourth one binding. It's called cooperativity. That's, I think, what's meant by this. So hemoglobin actually has exposed in basically 4 more different conformations, because as each oxygen binds, the whole structure of the 4 units undergoes changes in its shape. So actually it's probably, I think there'd be more than 4, it's probably multiple conformations as you see each incremental state of atoms and bonds changing directions, so orientation. So the AlphaFold has a limited success with predicting these, different conformations. I think that's what that refers to. AlphaFold 3 can predict structures of complexes with very limited selective. Yeah, I mentioned it's when you have things like ATP or ADP or various ions or NAD, various metals required to be bound, like zinc and zinc fingers, or iron in hemoglobin or magnesium in chlorophyll. It's a 50 limit of success. 50 to 70% of the structures of human proteasome are incomplete. That's not meant to happen. Are incomplete. Also without covalently attached sugars, glycans. Alpha fill is another thing it's required to fill in the missing bits. And also what I read is this is saying the algorithm, the residues are freely to move without constraints, so you do get integrity of the chain is not maintained. So I mentioned this earlier that you do get protein structure predicted where it's actually topologically wrong, so that's the shape of the geometry is incorrect. So all the predicted structures require crystallography backup confirmation. I think the Nobel Prize is overhyping this work. I can't really see the point of it. For trying to make, as I said, trying to make a new protein that doesn't exist, you don't need to experimentally confirm it anyway. Although if you're designing a protein and make it, I mean, you probably would want to get crystallography data, I suppose, to check the shape. But I can't see the point in this. When I was on my PhD, what I was obsessed with was how does the amino acid chain fold up into the 3D structure. I wasn't interested in the 3D structure is interesting, but if you let's start off with really Linus Pauling when he predicted alpha helices and beta sheets from hydrogen bonding data. But I can't see the point in this work. I think the Nobel Prize is overhyping it, and it's been described as the holy grail of protein structure prediction. I don't know about that. I think protein folding mechanisms is a holy grail, and people like Alan First, Jane Clark, Sheena Radford, Chris Dobson are all the pioneers of that. There are other pioneers of protein structure, people like Dorothy Hodgkin, Max Perutz, John Kendrew, and in more recent times you've got all these protein folders, but Richard Henson, there's a lot of Carol Robinson. I think it's overhyping it. I don't know. I just think it's I think the Nobel Prize is overhyping their work, and I can't really see the point of it. And it's the cells already make proteins. I think I just don't know. Oh, so somebody was telling me, a professor said, it allows you to predict thousands of structures because crystallographers just can't get through proteins quick enough. That's not really the way science should work. It's not about making thousands of predictions that may be wrong. I think it's better to take your time doing individual proteins and make sure and work out the structures with X-ray and get them correct, not just spewing out structures in a computer, not knowing whether they're correct or not. That's not good science. So anyway, this is my thoughts on it. So other limitations of AlphaFold 3 are something called chirality, the molecules of chiral. This is alpha chirality. It's not always respected with the AlphaFold program AI. Not always respected. What chirality is in a nutshell is when you have a central atom, say carbon, and you have 4 groups coming off it. So it means that it kind of creates a new kind of set of isomers. So if you make another one, if you make these 2, if you make, this is a chiral atom, okay, a chiral center. If you have 2 molecules a bit like this, but you swap 2 of the atoms around, then whichever way you put them, they are not superimposable. So they basically are isomers. They're different. If you can superimpose them, if you can take 2 of these and you can superimpose them, it means they're not chirals. It means they're not chiral, they're not isomers. Sorry, yes, they're not isomers. So that's what it means. It's basically having a center where the 4 atoms are different, so you get different isomers, and they're not superimposable. So that's, I'll do a video on it properly some other time, but that's a simplistic summary. And it's not a simplistic summary, that's a very brief summary of it. It's central atom with 4 different groups, and it means they're not superimposable if you have 2 molecules like that, and if they're not superimposable, they're basically isomers. If they are superimposable, they're not isomers. So chirality is not always respected. Sometimes you get clashing atoms and even chains overlap. Okay? Lots of the clashes occur most when you in nucleic acid protein complexes. So for protein complexes, when you get greater than 1,000, greater than 100 nucleotides and greater than 2,000 amino acid residues in total. So this is still a problem. It produces static structures, like with protein data, the protein data bank. So when you take a crystal structure of protein, they're called PDB files, protein data bank files. So AlphaFold programs produce static structures, not dynamical structures. When you run a simulation, you get dynamic structures. So this is clearly not a simulation. So AlphaFold is not a simulation. I'm not quite sure what the fuss about this is, because when you build a protein structure in crystallography, it's a static thing, it's not moving. The simulations only occur if you have to put Newton's equation into a computer, into the software, and then you put your PDB file in with all the atom coordinates, and then you put Newton's equations in, and the equations of motion, and then when the program calculates it all, it basically makes all the atoms move by Newton's laws of motion, by Newton's mechanics. I don't know, you know, they obviously, maybe they're trying to build this up to be able to do simulations with it, but this is more of a fact rather than a limitation, but because all the structured pioneers have never been simulators. Anyway, and then you got ligases, for example. Sorry, when you have ligands attached, for example, iron or ATP, they have to be added later. In, for example, E3, ubiquitin ligase, which is a protein that makes bonds. In that case, it would make them. This has an open native conformation in its apostate and closed in enclosed with ligands. So aqueous state is when there's no ligands attached, closed with ligands. So yeah, just so it's closed with ligands attached, so clearly the AlphaFold 3 system is flawed because it predicts both closed states of both the state with ligand attached and without ligand attached, so it's clearly flawed, this AI. And the last example is when you have antibody-antigen complexes. Antibodies are the immune system, and antigen is the foreign body, okay? So that's the Y-shaped molecule that attacks foreign bodies, and this is the foreign body, okay? So these are hard to predict. Things when you're doing crystallography, the old-fashioned way of doing this thing, crystallographers have successfully made antigen antibody crystal structures and enzymes and proteins and all kinds of molecules. So the old way of doing it with crystal, or the standard way of doing it, say using X-ray, works fine. Clearly AI, DeepMind, and this AlphaFold 3 software they created, however, I don't know how it works, I presume it's software, is flawed. It's not succeeding in making a lot of these structures. So it's not succeeding in predicting protein structure and protein complex structures and protein nucleic acid structures successfully. So I think it's overhyped. As I said, I think it's probably didn't really deserve a Nobel Prize. A professor I know has agreed many Nobel Prize winning things as the gimmicky. I do think gimmicky. I think often it's because often things don't win Nobel Prizes, which are actually major breakthroughs, are important or interesting, and often things that do win them, you kind of wonder why they won them. Yeah, but at the end of the day, they have to be checked by experimentalists, by the crystallographers. And then when they did during COVID, they did the spike protein and some of the other COVID proteins, but they had to send them to the Francis Crick Institute for verification. Their models, the model they predicted was correct, but I was actually in a protein folding PhD 20 years ago, sorry, and I studied protein structure in my degree in a lot of depth, like you do in degrees, and I can't really see the point of this AlphaFold, this DeepMind protein prediction. I can't see the point of it. Our body cells, animal cells, body cells, plant cells, they make proteins anyway. I thought the goal of trying to figure out how proteins get folded up into 3D structures was more interesting. To me, the predicting protein structures, although they talk about how it helps them develop new drugs and coming up with new protein structures, coming up with new protein structures doesn't require predicting a protein structure. If you're trying to invent a new structure, I don't see how that requires having to predict it, because the protein doesn't exist anyway. But if you're trying to come up with the structure of, say, hemoglobin or something, or some other enzyme, and you want to check whether it's correct, you have to verify it against experimental evidence, or against experimental crystal structure. I think this whole Nobel Prize is overhyping this work. I think it's certainly not as good as it. It's not the AlphaFold is not very good at determining. It's got low success at working out the structure of disordered parts of proteins. It also has not been so great at figuring out where things like ATP bind or ADP bind, or various ions bind. So in hemoglobin you got iron, in metalloproteases you got transition metals, metalloproteases enzymes. In what do you call it? Zinc fingers you got zinc and zinc ion. It's apparently earlier versions have not been very good at knowing where those go. AlphaFold 3, I think, is better, but they have to rely on something called Alpha Find, whatever it's called, to Alpha Fill to fill in these details, these extra bits. So, yeah, the